We're here today with Laureate Professor John Aitken at Newcastle University in Australia. John, thanks for your time.
My great pleasure.
Talking a bit closer about the Felix device,
Yeah.
can you explain, probably in layman's terms, how it works and why it's important?
So my involvement with Felix really started actually shortly after I got to Newcastle, and one of my first tasks in Newcastle was to set up a degree in biotechnology with clinical and industrial placements associated with it. So I got to visit a lot of biotech companies in Sydney, and one of those was a company called Gradipor. And I became good friends with Tim Warren, who was the CEO of Gradipor at the time. And they built a device, the Gradiflow device, which was used to separate molecules on the basis of size and charge. And I suggested to Tim one day that we could use this technology to separate cells. So we built a prototype device called the CS10, and I said, I think I have the perfect cells to try this on. And so we'll try it to see if it can effectively separate human sperm cells, because the IVF industry needs a mechanism to separate good cells. And so it was, and basically it separates these cells on the basis of their unique size. They are the smallest cells in the body, and their high net negative charge. So it's essentially an electrophoretic technique that isolates the cells on the basis of their small size and high electric charge.
How do you guarantee that DNA is not compromised by using this technology?
Yeah. So one of the attributes of this particular technique is, unlike conventional techniques, we don't use any centrifugation. So the way you usually isolate cells in IVF programs is to centrifuge them. These involve shearing forces as you isolate cells on the basis of high G forces. Our technique involves no addition of no chemicals, nothing to the cells. It doesn't involve any shearing forces associated with centrifugation, and it's very quick. Unlike centrifugation or another technique which is often used, which is swim up, it takes a fraction of the time that those techniques take. And the cells that it's isolating are those with the highest net negative charge, and those are the cells which have the least levels of DNA damage within them.
So moving forward to a user case example, how would you see it used in an andrology centre?
Yeah, so the first time we ever used it was when I received a phone call from actually Sydney IVF, who had a patient who had been infertile for about 10 years, tried many different times to have IVF, never worked. He had high levels of DNA damage in his spermatozoa. So we took our prototype device down and isolated cells from him. The subpopulation we isolated had very low levels of DNA damage. We collected those cells, injected a single spermatozoon into one of his wife's eggs, and she immediately conceived and had a child, and subsequently had another child using exactly the same technology. We were encouraged by that pilot study, and we then did a much larger clinical study at Westmead Hospital. And in Westmead Hospital we selected quite a large group of patients and used this technique and also the conventional method of isolating cells, and then just selected the best embryo for transfer irrespective of how it was prepared. And the outcome of all of that was to show that our technique was very effective, at least as effective as conventional methods for establishing a pregnancy. And in fact, pregnancy rates were higher with our device than they were with conventional methods, although the study wasn't really powered to say that with confidence. Nevertheless, pregnancy rates were higher. So it's at least as good as conventional technologies and takes a fraction of the time.
So just touching on that, why would an andrology centre use this technology?
Yeah, because I think as a matter of best practice, andrology centres should select cells with the lowest levels of DNA damage. There's quite a lot of data out there suggesting that high levels of DNA damage are not good either for pregnancy or for the health and well-being of the offspring. So we should, as a matter of best practice, select the cells with the lowest levels of DNA damage. And this technology, this new technology, enables us to do just that. It's much better than conventional technologies and takes a fraction of the time.
You look very excited and you look proud.
Yeah, I am very proud of it. It's a device that has been a long time in the making. It's not been a linear journey. often the case. We've had many twists and turns, many ups and downs. But now I think we're in the best place we can possibly be. We've got a good network of people who are working with us to evaluate the system. We've got it in the hands of a company who are passionate about the device, as passionate about it as I am. And I think if this device has any potential for commercialization at all, it will be realized now. Now is the moment for that assessment.
In terms of market size, given your breadth of knowledge about the industry from a human perspective, what do you see as the opportunity here for the Felix device?
So I think the potential for this device is immense. ART, assisted reproductive technologies, are now being widely used across the world actually to produce IVF children. Currently in Australia, 5% of all children are generated by IVF. In Denmark currently 10% of children are generated by IVF, and in all developed countries the vector is upwards. So it's 5% now, by 2025 it'll be 10% and so on. And there are very good reasons for believing that this is just going to continue to grow. There are already 8 million IVF babies in the world. The technology though has remained unchanged since it was first introduced in the late 70s, early 80s. So I think now is a good time for us to revolutionize the technologies that we're using for IVF. Not so much to increase efficiency. I don't make any claims for increased efficiency. What I am interested in is the safety of this particular procedure. And the fact is that with IVF, unlike natural conceptions, the gametes, the sperm or the eggs are not selected in the way that they are in vivo. So with this device, we're able to select the highest quality sperm for conception. And that will ensure, I think, the health and well-being of the offspring that are generated. And that's a matter of supreme importance to me and I think to the industry itself.
Do you believe this device will be the standard device that sits in every andrology centre based on the outputs you just mentioned?
Yeah, no, I think absolutely that's going to be the case. So as I've already said, the technologies have remained unchanged really since the inception of the IVF therapy itself. And it's about time that they did change. This method enables you to isolate very high quality of cells in a fraction of the time that conventional techniques take. So I don't see why this shouldn't be the default technique that all andrology laboratories use in the future for isolating cells for IVF. This will become just part of the standard protocols that are used. I think this technology and other technologies relating to selection of highest quality embryos are the two technologies that are going to carry this whole field forward in the immediate future.
Was that little glimpse generated by how eager the KOLs were to be part of the study?
Oh, absolutely. I mean, this is, you use the right word there, it is a collaboration, it's an intellectual and scientific collaboration. Because I think my colleagues can see how this technology will vastly improve the way in which we isolate cells, high quality cells, for assisted conception purposes. And ultimately, you know, our ambition is that everybody who goes home with an IVF baby knows that we have done everything possible to ensure the quality of the gametes that went into making that child. That the health and well-being of the children generated through ART is of the highest possible quality. And this technology will enable you to achieve just that.
Professor John Aitken, thanks for your time.
My great pleasure.